Yep. Okay. Great. Oh, well, thank you, Yvonne, and thank you everyone for making the time in your schedules to be here and to witness what I'm about to share with you. I'm very excited to get started. Okay, so the title of my dissertation, Assessing Biodiversity in Madagascar Through Leech-Derived iDNA, Methodological Advancements and Ecological Insights. Before we get started, I do want to issue a gentle warning in all seriousness. If you have an aversion to blood, I am talking about blood-feeding leeches, and there will be pictures of leeches and of blood in my presentation. So what is biodiversity and why does it matter? Biodiversity is simply the variety of all plants and animals on Earth. And this variety helps maintain clean air and clean water. It helps regulate our climate and buffer the spread of infectious disease. It's also a source of medical and pharmaceutical discoveries. And tragically, we see across the world that vertebrate biodiversity in particular is in decline. And to safeguard against this decline, scientists need baseline measures of biodiversity to monitor trends over time. Traditionally, this has been measured with camera traps, as you see here, which are just cameras fastened to a substrate.
Hi.
Yes.
Sorry, you haven't shared your screen.
Oh my goodness.
That's okay.
Okay.
It's a great intro.
Wow. Hello. Welcome, everyone. Okay. Sorry about that. So we were just talking about biodiversity being the definition of just the variety of all life, animals and plants, and it's in rapid decline across the world. It helps maintain these very crucial services. And traditionally, to safeguard against this decline, scientists usually use camera traps, which you can see here are just cameras fastened to a substrate. More recently, researchers have begun targeting blood-feeding invertebrates like flies and mosquitoes as sources of biodiversity DNA sourced from within their gut contents. And researchers have also begun targeting leeches for this new technique known as invertebrate-derived DNA. Now, when it comes to leeches, they perform better than other blood-feeding invertebrates because they have been shown to preserve high-quality DNA in their guts on the order of months. The leeches that I work on and the ones that I'll be discussing today belong to the family Hemidipsidae. They are the terrestrial blood-feeding leeches. They're also known as jungle leeches. They are abundant and extremely easy to collect. They're also phylogenetically differentiated by site. And so they exhibit, we've seen some evidence that they can exhibit site fidelity, which means that the blood meals retained from those leeches will reflect the localities from where the leeches were collected. And this isn't necessarily the case for flying invertebrates, which do tend to travel far distances. Here I show the global range of the terrestrial leeches. They're found across the Indo-Pacific, which is a region that harbors some of the world's most endangered wildlife. So seen here in red are the world's biodiversity hotspots, and these are regions which simultaneously harbor high species richness, high species diversity, and are also extremely threatened with extinction. And among these hotspots, the hottest often seems to be Madagascar. Up to 90% of the original forest cover has been lost due to logging in Madagascar. A majority of the species are endemic, meaning they're found nowhere else in the world. And most of these species are relying on rapidly shrinking habitat. And so this makes for a compelling system where leeches are abundant and species are highly diverse and endangered to test the use of iDNA in Madagascar. And so I wanted to give you an idea of how this works. So on the ground, here I am, me and my team walk slowly through the forests in Madagascar, collecting leeches as we find them. and we put them into these little collection bags, making sure not to collect any leeches that have latched or fed or broken the skin on any human researcher, seeing as we're not interested in recording the presence of human DNA in these forests necessarily. And the samples are brought back to camp where I sort and process them roughly by color and pattern, and I get them all labeled and packaged to get back into the lab where I individually dissect each leech for this region, the posterior crop region, which contains the blood meal. And to give you an overview of the process as a whole, the first step in analyzing leech gut contents is DNA extraction. Now, it's important to realize that leeches have their own DNA and that in the iDNA approach, I'm targeting specifically blood meal DNA, which reflects the identity of the hosts of the leeches. Oops. So the next step is PCR amplification, which is basically making many copies of target genetic regions. And there are short regions of DNA that have differentiated really highly across species, and they effectively serve as barcodes, much in the way that we can scan a barcode to look up information about a product. We can target these regions of DNA and make conclusions about the species identity of hosts. Next in this procedure is DNA sequencing, which is just the process of determining the order of nucleotides in a given genetic region. Those sequences are then queried against a massive global database containing DNA sequences representing hundreds of thousands of species. And finally, we get a match from the database, and as species are identified, we're interested in their IUCN categories, the IUCN being the International Union for the Conservation of Nature, the body that oversees the categorization of species endangerment. And so often while I'm sharing my results, you'll see next to the species name, I've included the abbreviation that stands for each category of IUCN endangerment. So broadly, I ask, can leech-derived iDNA be used as a reliable biodiversity inventory tool? In chapter 1, I use micro-CT imaging to describe a new species of leech. I also scan previously described leech species and reconstruct their internal anatomy visually to compare against these prior dissections, which are traditionally how leech species have been identified. As Yvonne mentioned, when I first started my research in 2017, iDNA was really in its proof of concept stages, and the standard approach was targeting a single genetic marker with traditional Sanger sequencing. And so in chapter 2, I take it one step further and I target 2 genetic markers, one specifically designed to detect mammal DNA, that's the 16S region, and the other designed to detect vertebrate DNA more generally, and that's the 12S region. And I approach this technique with Sanger sequencing. In chapter 3, I take a massive methodological leap comparing blood meal samples that I combine into pools. And I add 2 more genetic markers on top of 12S and 16S, that's ND2 and CO1, targeted for the detection of birds and reptiles respectively. And I use next generation sequencing, which is a more advanced sequencing technology to try to optimize this iDNA technique. Finally, in chapter 4, I apply the iDNA method in a newly described and undersurveyed forest. I test one more methodological approach where I combine leech tissues before proceeding with analyses, and I compare the results of my iDNA survey to the results of a survey conducted by a research team in Madagascar belonging to the Centre Valbaya Research Station in collaboration with Stony Brook University, which use standard methods like camera traps, bioacoustic surveys, and visual surveys. And so throughout the talk, you'll notice me referring to data collected by CVB. You can think of this as sort of a proxy for more traditional biodiversity monitoring methods. My 4 chapters take different approaches to look at the utility of the iDNA tool. And the work that I'm presenting today has helped establish this technique as a valuable biodiversity monitoring method in the tropics of the Indo-Pacific. And it's a journey of my own professional development and growth as a scientist, and I'm really excited to take you along. So I collected each data for each chapter from these respective forests across Madagascar. Chapters 1, 2, and 3, the data for those chapters was all supplied from Ranamafan National Park. The data for chapter 4 was sourced from Andasi Bay, Ranamafan National Park, and the Ivory Protected Area. So my 4 dissertation chapters are listed here. I did want to make a note that a dissertation is more like a network of projects that aren't necessarily completed in order. And so with that in mind, I'll be presenting my chapters to you today in the order in which they were completed. And so I start with chapter 2. Biological inventory of Ranamafan National Park tetrapods using leech-derived iDNA. In chapter 2, I analyzed the gut contents of leeches collected from Ranamafan National Park by increasing target genetic regions to include the vertebrate-specific 12S region, in addition to the more standard mammal-specific 16S region. I also used the traditional Sanger sequencing approach. This chapter has been published in the European Journal of Wildlife Research, and I'll be summarizing my findings for you here. For more detailed results, you can look at this publication. I begin my sample collection in Ranamafan National Park, a large park of 43,500 hectares. To give you a bit of an idea, Central Park is about 340 hectares, a hectare being just under, a little bit under an acre. The park was established by Dr. Patricia Wright at Stony Brook back in 1991. So this provides decades of biodiversity surveys against which to validate iDNA results. So this was a good place for me to start my research. There are also regions of selective logging throughout the park that have created sort of a gradient of habitat disturbance, allowing us to ask questions like, how does biodiversity vary by site? So to collect leeches, me and my team hiked 200-meter transects perpendicular to established trails throughout the park, and that was to try to avoid human DNA contamination as much as possible. We stopped at every 50 meters to inspect ourselves and our clothing, collecting basically any leech that we could find, so long as it hadn't fed on one of us. We collected from 3 sites, varying in degree of disturbance, where I defined disturbance here in terms of proximity to the main road, which you'll see at the top of this map in yellow. So Talatakeli is the site that is right up on the road. It's also our site of heaviest disturbance. Mangevu is the site that is furthest from the road and is our pristine site. And Vatu sits between the two and serves as our site of intermediate disturbance. In this chapter, I extract DNA from individual leeches and I keep each blood meal separate. At the time of this work, targeting the 16S mammal-specific genetic region was the standard protocol for the newly developing iDNA method. I take it a step further here by also targeting the 12S region, which, as I said, is a more general vertebrate-specific marker. And I sequenced these target regions after PCR amplification, which again just means making very many copies of each region. And I sequenced this DNA with Sanger sequencing. So once again, DNA sequencing is just the process of determining the order of nucleotides in a given genetic region. The Sanger method can only sequence 1 genetic region per sample. And usually it requires high concentrations and high quality of starting material. So it's usually better suited for non-degraded DNA, but it is great for targeting a limited number of genetic targets. And because DNA sourced from leech guts is inherently degraded, right? It's sitting in inside the digestive tract of a leech, being slowly digested. Sometimes there isn't enough resolution in the DNA samples to identify hosts to the species level. And so often you'll hear me refer to my results as reflecting family level diversity. And that serves to capture a broader, more conservative estimate of diversity. So species include naming with genus and species. Family is a broader classification of diversity. So this is partly because the DNA is degraded, but also partly because this reflects the availability of sequences in the database. And so any method that relies on a reference database is going to rely on the completeness of that database, or that the database is sufficiently populated with species relevant to your research system. And so if there's an exact match in the database to a sequence that you've queried, you'll get a hit to that match. And if there isn't, you'll get a less precise hit to the next best match. So to try to remedy this issue as much as possible, I supplemented the database that I'm using, GenBank, with Malagasy endemics that weren't initially represented on GenBank from the American Museum of Natural History's frozen tissue collection. And so here are some of my first results comparing matches from the database to the 16S or mammal-specific region in blue to using both 12S and 16S represented in green. And here I just show raw sequences matching to endemic mammals, birds, reptiles, and amphibians as determined by genetic region. And overall, I see a 41% increase in host species identified when we target both genetic regions. I also find that 100% of amphibians were detected exclusively with the 12S region. Here I summarize respective proportional vertebrate diversity by class using both the 12S and 16S identifications as they correspond to collection locality from across Ranamathon National Park. So we see here more birds are detected in Talatakeli, more mammals in Vatu, and higher proportion of amphibians detected in Mangevu. I also conducted a chi-square test to test the frequency of detection of taxonomic classes across our 3 sites. And I do find that there's a relationship between locality and the proportion of species detected. And so where we see more birds in Talatakeli, this is actually a pattern that's been observed that suggests that birds are found in higher abundance in degraded forests. And so it's interesting that with iDNA, we're able to pick up on these differences in community composition. Now, because of their previous absence on GenBank, the supplementary sequencing I had done from the AIM&H frozen tissue collection allowed me to identify these species here, which would have been otherwise missed if I hadn't supplemented my database. I also present the first evidence of terrestrial leeches feeding on skinks and snakes, showing that leeches are able to latch and feed through tough scales. And so some of my main takeaways for this chapter are that adding 12S to 16S as an additional genetic marker helps increase host species identified. The 12S region is particularly effective at detecting amphibians. And overall, the iDNA results from this work seem to detect mainly small ground-dwelling species, and those tend to be species which are also too small to trigger camera traps and are also just generally easy to miss. Chapter 3, multi-locus metabarcoding of terrestrial leech blood meal iDNA increases species richness uncovered in surveys of vertebrate host biodiversity. So here I use the same samples from chapter 2 collected from Ranamathon National Park to test the addition of more genetic markers, so that's ND2 and CO1, to the previously tested 12S and 16S regions, which I tested in the previous chapters. I also combine blood meal DNA in these samples instead of analyzing individual leeches, and I do that in a way that's meaningful. So I first separate samples by leech species and then by collection locality. And because we're not necessarily interested in keeping these data separate because we want an overview of the ecological community, we're not so concerned with the contents of individual blood meals per se. I also use different DNA sequencing technology, and that is next generation sequencing, which I'll get into a little bit. The results of this work are published in the Journal of Parasitology, and I'll be sharing some of my findings with you here. My collaborators on this paper contributed data from terrestrial leeches that they had collected. So those leeches were sourced from China, Bangladesh, and Cambodia. So in addition to the leeches that I collected from Madagascar, I'm also testing these new methodological approaches on those samples as well. And in general, increasing sample size helps us see patterns in the data more clearly. So I was really excited and eager to incorporate their samples. So overall, we're targeting over 1,200 leeches with these new approaches in this chapter. And so, as I mentioned, because I'm not interested in knowing the host of each individual leech, but rather the diversity of the community, I combine or pool blood meal DNA samples based first on leech species and then by collection locality. This reduces the total number of samples that I'm dealing with, and it helps save time and reagents. And I also use next generation sequencing, which is technology that's highly sensitive. It makes it really great for degraded samples. You only need a small amount of starting DNA material, and it produces more data than does the Sanger sequencing approach, because it's able to sequence multiple genetic regions per sample. So I just wanted to take you along and show you what this pooling process looks like. So we pool our samples to create these templates that we use to target these genetic regions. And each one of these barcodes is optimized for the detection of birds, reptiles, mammals, and vertebrates more broadly. Of course, all of these are vertebrates, but using a specific barcode enhances species identification. And what we get back from next generation sequencing are millions of sequences, but not all of them are usable. So we remove them based on size and quality. And we also remove redundant sequences. We then cluster them, the remaining sequences at 98% similarity. And we filter these clusters to get rid of non-vertebrate DNA contaminants, things like plant and bacterial DNA. Finally, the surviving clusters are queried against GenBank's non-redundant global nucleotide database. And we get our results. And so in the case of this frog, for example, at 98% identity, we can be relatively certain that this is Gepharmantis rhytomitis, whereas at a lower percent identity, we might only be able to determine that it's a frog. And here I wanted to show a comparison between the next generation sequencing results and the Sanger sequencing results of just the Malagasy samples of just the leeches collected from Madagascar. And I show that with next generation sequencing, I find an additional 10 species level identifications from the same set of data and 14 additional family level identifications. And these are just some candidate representatives from the new hits with next generation sequencing. Here I show the proportions of family level identifications by taxonomic class across all of our data. So that's from leeches sourced across Asia as well as Madagascar. And on the left, we see that birds and mammals constitute the most diverse groups across my sites. We also see that they constitute the highest proportion of ambiguous taxa on the left. And so where I refer to ambiguous taxa, what I mean is if the top hit and the second hit to the database belong to different families, but the same order, they're represented on the right as being ambiguous because I can't really get to that family level resolution. And so this is due in some part to the incompleteness of the database, but also to the fact that birds and mammals also constitute the most diverse groups across these sites. So this makes their diversity inherently more difficult to represent on databases. Here I just show how well each genetic marker performed, and based on these frequencies, I find that ND2 and CO1 increased species detections by 25%. Now, if a study of this scale with over 1,200 samples and 4 genetic markers was attempted with Sanger sequencing, which can only sequence individual samples at a time, it would cost over $30,000. Whereas with sample pooling and next generation sequencing, we're able to reduce costs by 85%, coming out to roughly $4,400. Time spent in the lab would be roughly 110 days to try to approach this work with the Sanger method of sequencing. And with next generation sequencing and pooling samples, we can get that down to 17 days in the lab. So the next generation sequencing approach really seems to be a more effective and efficient approach. One interesting result was the detection of this invasive amphibious fish. which actually is able to travel short distances across land. And again, this is the first evidence of terrestrial leeches feeding on fish. And so some of our main takeaways here is that next generation sequencing pooling samples and using multiple genetic targets is going to increase host species identified while also reducing costs. iDNA really detects a wider breadth of taxonomic diversity than is typically known from other methods. And it does show some promise of detecting and geographically limiting invasive species like this fish. Now, chapter 4, leech-derived iDNA complements traditional surveying methods, enhancing species detection for rapid biodiversity sampling in the tropics. This brings us to one of the culminating events of this dissertation, where I had the opportunity to collect leeches from the newly described Ivohiburu protected area. I was invited to employ my iDNA technique because the site at the time was still being vetted for protection. For this chapter, the data were supplied from Andasi Bay, Ranamafan National Park, and the Ivohiburu protected area. Now, in 2016, researchers affiliated with Stony Brook and Center of Albio received a call from a Malagasy woman who was claiming that a small rainforest in her hometown in south central Madagascar contained ring-tailed lemurs. Now, this was sort of surprising because ring-tailed lemurs are typically only found in dry forests, and they're typically usually ground dwelling, and she was reporting them living in the trees in a moist rainforest. So this sparked lots of curiosity in the scientific community, and several expeditions went out to determine what exactly lived in this small fragment that had been protected from the recurring human-induced fires by very steep cliffs. It quickly became clear that there was an unusual community of wildlife in this small 3,300 hectare forest, and I was part of some of the first scientific expeditions to the site in 2019. The CVB team conducted surveys over the course of 3 years and 6 expeditions, totaling about 90 days of data collection, where they were exclusively interested in trying to document as much diversity as possible across the site. And they used a suite of methods, conducting surveys during the day and in the evening. They recorded the sounds of the forest for bioacoustic monitoring. They used baited traps. And so it was really a massive surveying effort that they conducted. And they find that there does appear to be a mix of wet and dry forest species that aren't usually found together in Madagascar. And so how did this unusual mix of species come to live in this forest? Does the composition of the Ivohiburu protected area reflect a remnant community of older intact forests? Or have species expanded their known ranges into the IPA in search of refuge? Answering this particular question is outside the scope of this chapter, but approaching this question requires that we first understand exactly which species are found in this fragment. And so here I use iDNA methodology optimized in chapter 3, and I compare my iDNA survey results to those carried out by the CVB research team, again, using cameras, bioacoustic surveys, and visual transect surveys. And I also take the sample pooling a step further, and I test how pooling leech tissues may influence species identification. This publication is ready for submission pending our collaborators' return from the field, and hopefully you'll be able to read it soon in the journal Conservation Science and Practice. So I compare 2 approaches in this chapter. On the left, I combine leech tissues and I extract blood meal DNA from these samples collectively. And on the right, I combine blood meal DNA extracts of the same leeches, and I analyze the number of host species identified with each technique. These leeches that were used to test this pooling protocol were sourced from Andasi Bay, where I had an opportunity to collect. And I find that pooling leech tissues was more effective than pooling DNA extracts, which didn't return very many high quality species identifications other than to that of human DNA. I was part of the first ever scientific expedition to this fragment in 2019, where over a period of 12 days with a team of 2 people, we collected 1,451 leeches collecting along 200-meter transects, a similar strategy to that of the collection method that I used in Ranmafan. And I show here the family level detection. So on the left, we find that both CVB and iDNA surveys detected 14 families in common. I also find that iDNA was able to detect 6 families that the CVB team did not. Now on the right, we have a graph that represents host diversity as a function of sampling effort, and we see fewer new species detected as sampling effort increases. And so at about 1,451 leeches, it seems that we have nearly captured the full extent of host diversity possible with iDNA. Here I show the additional families detected with iDNA. Of course, 2 of these families are domesticated species, and we see them because there are villages that exist around the IPA, and many villagers use this fragment to graze their cattle. I don't count the ambiguous hit to a cat as a domesticated species because the Malagasy people in the region do speak of a forest cat that is different from a classic stray in the region. And so this may be what we've picked up here with the leeches. But yet again, we do see that iDNA effectively targets small ground-dwelling species that are often missed with traditional surveys. When we compare family level detections between the iDNA results and CVB results, we see that iDNA is best paired with other methods, especially when we look at our side-by-side results among birds, which again offers further evidence for iDNA's ability to detect mostly ground-dwelling species. Here I show the proportion of families categorized by taxonomic class representing the diversity detected with just iDNA. And I have here predominantly hits to mammals and birds. I also wanted to share some of our species level identifications. So I was really excited to see that we got hits to a chameleon and also the endangered lemur cata that brought us to this site in the first place. It was also really exciting to see that we find 4 frog species that were not accounted for by the CVB surveys. And so some takeaways here are that pooling leech tissues prior to DNA isolation effectively and efficiently targets host DNA. iDNA is a complementary method to more traditional surveys. And the next steps at the site are going to be to analyze and observe relationships between the populations found within the IPA and from across other forests in Madagascar. Now, let me take you back to chapter 1, where we look at a different technique used to assess the biodiversity of leeches themselves. So we're transitioning from looking at blood meal DNA to looking at leech DNA. And the reason that I'm presenting this chapter last is because the evidence of a potentially new leech species only really became apparent after I finished working on chapters 2 and 3 on the leeches from Ranmafan National Park. And so in this chapter, I use Sanger sequencing of a diagnostic genetic region, the COX-1 region, as well as microcomputed tomography, so that's micro-CT imaging, to present evidence of a new species in the leech, of a new species of leech in the genus Catanabdella. And so in addition to scanning the new species, I also scanned previously described leech species from Madagascar, whose anatomy had only been assessed in the context of dissections formerly. This chapter will be submitted to the journal Invertebrate Systematics. And again, now that we're shifting from blood meal DNA to look at the diversity of leeches themselves, I wanted to give you some more context on what leeches are and why we might be interested in studying their biodiversity. In the context of iDNA surveys, I want to be able to discern whether particular leech species have preferences for certain hosts. Leeches themselves are understudied organisms, and much of their relationships and diversity is still unknown. Leeches are parasitic worms that are related to earthworms. They contain both male and female reproductive anatomies per individual. As I mentioned in previous chapters, initially I was sorting leeches based on observations of external features. And here I wanted to use molecular approaches and morphology to see if these differences really represent any new diversity. So I build on some of the methods developed by my colleague, Dr. Michael Tesler, who's a Fordham alum, who was the first to use micro-CT scans to describe leech diversity in 2016. And so this is a tree representing genetic divergence among terrestrial leeches based on a diagnostic region of DNA, the COX-1 region. Species from Madagascar are labeled in red, and I represent a subsample of the leeches that I collected from Ranamaphon, shown with the indication of their sample size. And if you recall, I collected samples across 3 localities in Ranamaphon, and I noticed some patterns, particularly among the leeches from Mangevu, that I wanted to investigate further. And so I noticed some samples that are shown here in green seem to fall out into their own group, and they group apart from any other known terrestrial leech species from Madagascar. And so I take this to be the first clue that we might have a new species of leech among my samples. So MG and FG, those abbreviations stand for male gonopore and female gonopore. Leeches contain both male and female reproductive anatomies per individual, and the location of these gonopores is a diagnostic feature. I do find that they are separated by the same number of body segments as I find across the previously described leeches of Madagascar. So this spacing is consistent with other known species. When we look at the ventral patterning of this putative new species, I see that it resembles the patterning found in Cotanabdella phallax. And when we compare the dorsal patterning, we see that it resembles Cotanabdella vagans. And so we can't really stop there when it comes to trying to figure out whether or not this is a new species. External patterning alone cannot be used. And so traditionally, painstaking dissections on really tiny specimens were carefully performed to inspect the most diagnostic characteristics, and that is the morphology of the reproductive structures. However, with micro-CT, we can forego these dissections, and with micro-CT, the specimen is scanned with X-rays in 3 dimensions, and then with special computer software, those scans are merged and reconstructed in 3-dimensional digital space. So this helps maintain the structural integrity of the specimen, which is important for deposition in museum collections. Now, with volumetric rendering, which is basically coloring in the volume in 3 directions with tennis ball sized paintbrushes with a 3D paintbrush online, I've been able to show the internal structures of this new species is distinct from those of previously described leech species. So we saw that we have unique dorsal and ventral patterning. We also see here that proportionate to body size, the vaginal sac shown in pink extends the length of 3 body segments, which is larger than any of the other described leech species. And we also observe a distinct curvature of the ejaculatory bulbs seen here in blue. And these characters aren't seen in any other leech species. And for comparison, I scanned all previously described species from Madagascar to observe and compare internal anatomy accurately. And we can clearly see that this new species is different from the rest. And so some of our main takeaways here, we see that micro-CT scans accurately image the internal anatomy of leeches, and that these scans corroborate what we know about morphology from dissections. And as a side-by-side comparison here, I show the dissection of Cotanabdella phallax, which was from 2006, alongside my digital reconstruction with micro-CT, showing that the scans do corroborate the relative shape, size, and position of these internal features. Now, this is a development that's come about since I submitted my dissertation to my committee. But here I wanted to unveil the name of the new species. So that will be Cotanabdella mangevuensis, roughly translated from Malagasy. It means the bumpy swamp two-jawed leech. And when considering what to name something, we always want to take into account the local community and the journey of discovery. And since these leeches are from Manguevu, I thought I'd give you a glimpse into what it actually took to collect these samples. So our journey to Manguevu was not very straightforward. It was a 10-hour hike with lots of detours and, you know, got lost plenty of times, but finally made it. Had to cross a river to get to the site, hike treacherous mountains. Once we got to the site, I had an incredible team that I worked with, Fifi and Tumbu, who helped me collect over 500 leeches of their own free will. To keep warm, we sipped on traditional Ranu Napangu, which is Malagasy race water, took showers in this freezing river, shared my tent with my samples, and accumulated a lot of mysterious bug bites that were not leech bites. Manguevu was also the site where I started my leech collections, and so here, this is actually footage of my very first leech that I've ever collected. And as a quick aside, when I look at the leeches I collected from the IPA, from the Avoyburu protected area, I do see putative evidence of another new species, which is very exciting, and which is work that I'll be working on post dissertation. As a part of my journey working with communities of scientists and the people of Madagascar, I was invited to contribute a chapter on leeches in this massive endeavor. This publication of the New Natural History of Madagascar, which has recently become available to the public last month. Now, you may be asking, what is the point of doing all of this? How do biodiversity inventories really make any difference? Well, biodiversity inventories enable governments and stakeholders to prioritize the allocation of what are usually very limited conservation funds, and they allow them to funnel them where species and resources are most threatened. We find that iDNA improves our understanding of leech diversity and behavior as understudied organisms, and it's widely applicable not only across the Indo-Pacific, but also as of recently throughout the world, as other families of leeches, aside from the terrestrial ones, have begun to be targeted for iDNA analysis. Also, studying the biodiversity of forest fragments helps us to understand the effects of shrinking habitats on ecological communities and their resilience to increasing human pressures. As for my own next steps, I'm looking forward to continuing my work on degraded DNA from environmental sources, and I'll be carrying out biodiversity surveys in a national park, a massive national park in Egypt, working alongside communities to document this biodiversity. And I've received funding to carry out this work from Rolex and the Explorers Club. I have so many to thank, and the names listed here are only a select few. Completing this dissertation was more difficult than I could have ever imagined. And all of these people have helped play a massive part in helping me reach this milestone. And so, of course, I want to thank Dr. Yvonne Heckela for always being so enthusiastically supportive. She's patient and kind, and her interdisciplinary approach to research has really taught me how to ask big, creative questions. You're a role model for all scientists. To my committee, thank you for your feedback in this process and for your encouragement at each of our meetings. To my lab mates and my cohort, thank you. Shamin Sahibzada, one of my greatest joys is being able to discuss my research with you, my in-house scientist and biostatistician. I admire your genuine curiosity, and you just make me a better scientist. At a certain point, this dissertation took a really dark turn, and you've witnessed some of my hardest days. So thank you for always knowing exactly what I need. To my parents and my brother Omar, thank you for encouraging me to pursue research and leeches. And as difficult as it's also been somehow equally the most fun I've ever had. And I feel lucky to have found this career. And so I want to thank Doctors Sharon Cochran and Patricia Wright, who have mentored me from my time at Stony Brook. very early on in my research career. I started out studying earthworms in Dr. Cochran's lab, so I've always been loyal to phylum Annelida. Mariah, you make my life better in too many ways to count. Thank you for the unforgettable time we spent in Ranamafan National Park and for the few dozen hours that went into helping me put this presentation together. You understand me in ways that few people do, and I can't wait to be by your side when you defend your dissertation next year. And to CVB and the communities of Ranamafan, Andasi Bay, Ihusi, and really all of Madagascar, without their support and permission, I could have never set foot in a forest or collect a single leech. So really none of this could have been possible without support from the Malagasy community. And so I will be happy to take any questions at this moment.
Well, I'd like to go ahead and thank Mai for her presentation and give a round of applause. It was very clear, I think, to everyone what an endeavor this was. So hopefully, I believe I allowed everyone to unmute yourselves if you want to give a round of applause. We all missed being in the room together. What we'll be doing next is you can raise your hand if you're interested in asking a question, or depending on how many questions we have, you can go ahead and just unmute yourself and ask Maya a question. And once the public questioning is done, what we'll be doing is asking you all to leave the meeting, and then there will be a period of time during which the committee will discuss Maya's presentation with her in more detail. And hopefully we'll see you all at the end for a celebration. So, okay, go ahead. If you want to raise your hand, feel free to do so and I can call on you, or you're welcome to go ahead and just unmute yourself now.
Hello. Hello from the party room. Hi. Mai, amazing job. We have everyone here in the party room. We were cheering so hard. My question for you, I think you made a very compelling argument that leeches are important for the conservation of other animals. Is there any good reason to conserve leeches themselves, though?
So there have been some studies that show that leeches are actually really great indicators of forest quality because they require very specific environmental conditions to survive. And those happen to coincide with conditions that are created by forest canopies, so intact forests. And so in a way, keeping leeches around is a really good way to make sure that forests remain healthy, and that we can continue to use leeches to sort of help us make note of how forest quality is transitioning in the face of human pressures. Also, aside from their utility as potential indicator species of forest quality, they are no doubt sources of prey for smaller animals like frogs and birds.
Lucky frog. Thank you so much.
Mr. Weisenbeck, feel free to unmute yourself.
So
Yes. Oh, you're muted again. While we try to sort out the, oh, okay. The question is, when you find that 12S identifies more amphibian taxa than 16S, is that because your database is more complete for amphibian 12S, or the 16S primers are less compatible with amphibians, or something about amphibian genomes?
It could be that the amphibians aren't, that the 12S region isn't particularly, or that the 16S region isn't particularly well populated for amphibians in the database. I didn't do any work to supplement the database for amphibians, and so I take it to mean that they're better represented with 12S and GenBank.
Great question. Any other questions or comments? All right. Well, we have a lot more to do this afternoon, so thank you all so much for coming. We had a total of 55 participants at one point, which is a wonderful turnout. It's one reason Zoom makes me happy sometimes, despite its unfortunate origins and our use for dissertations. So I'd like to thank everyone for coming, and I will be in touch after the private questioning period, and please enjoy the beautiful sunny afternoon wherever you are.
Thank you, everyone.
Woo! Don't leave, we wait, right?
Everyone leaves. You're all free to leave the room now, and we will follow up with you after. All right, I think you turned the recording off, but I can't tell. So.
Oh no, I don't think I have. Hold on, let me do it again.